Here is an oral capsule approved in April 2023 by the FDA and is administered to a patient 4 times a day for 3 consecutive days to restore their gut microbiome and prevent recurrent Clostridial difficile infection after a antibacterial treatment. But here's the thing. Inside this oral capsule is 9 bacteria spores purified from healthy human fecal matter. And what I just described is Fecal Microbiota Transplant, or FMT. It is a procedure that transfers processed screened stool from a healthy donor into a patient's gastrointestinal tract to restore healthy gut bacteria, and it has been shown in numerous cases to have high success rate in curing recurrent C. difficile infections. But here's the million-dollar question: why does it actually work? It's not just magic, it's a numbers game. To prove why it helps, researchers use advanced statistics to analyze the who and what of the trillions of microbes being transferred. Today, I am breaking down the statistical method used to analyze FMT. The first metric is alpha diversity. This measures how many different species of bacteria live within a single sample. So, for the statistical and the analytical side of things, the researchers use the Shannon index or observed features to calculate this diversity, and a lot of research had indicated a successful FMTs shows a significant increase in the Shannon diversity, meaning a more stable, healthy gut ecosystem. But it's not about having more bacteria, it's about having the right bacteria. For this, the researchers look at beta diversity, which compares the overall structures of communities. So for the statistical side, the Bray-Curtis dissimilarity is the go-to metric here in visualizing how different the two samples are. And researchers use the permutational multivariate analysis of variance or analysis of similarities to test if recipients' microbiome has shifted closer to the donors. In a successful transplant, the recipient's community structure changes from dysbiotic to resembling the healthy donor. And to get more specific, which exact microbes did the donor provide that took over? And researchers use the differentiational abundance analysis. This method involves algorithms used to normalize the data and find which bacteria are significantly increased in successful recipients. And the goal is to identify the biomarkers of success. And the key finding will show a high relative abundance of beneficial short-chain fatty acid producing bacteria, which is associated with clinical remission. To summarize, this statistics had shown that FMT helps because it resets the recipient's gut microbiome, which had increased in diversity. And secondly, it engrafts donor strings, so the recipient's gut becomes similar to the donor. Finally, FMT helps to fight pathogens and replaces it with beneficial bacteria. Analyzing FMT is a rapidly evolving field, moving from simple relative abundance to incorporating functional multi-omics. By understanding why it works, it can help to improve donor selection and make this therapy even more effective. Thanks for watching and stay curious.